We will be waiting 5 more minutes for more people to join, and then we will start. Good evening, everyone. I hope I am audible. Please let me know if my screen is visible when I do this. Okay, is my screen visible?
Yes, sir. Yes,
Okay,
sir.
okay. Good evening, everyone. Welcome to the week 5 PMRF-NPTEL session on animal physiology. In this particular week, we have dealt with the videos related to neural system part 2. In this, we have dealt in depth into how exactly neurons function, about various parts of action potential as well as membrane potential, and how exactly the ionic balance is kept across the neuron so that they may maintain a membrane potential. So this is what we will be discussing today as well, and we will be learning to answer many questions regarding this. So if you have been here in previous sessions, you would know that how we do this is that we will be going through various assignment questions from the previous year's run of animal physiology on NPTEL, and then we'll be trying to answer this question and discuss afterwards. I will briefly introduce myself. My name is Anubhav Chatterjee. I am a Prime Minister Research Fellow and PhD Scholar at IIT Kanpur in the Department of Biological Sciences and Bioengineering, and I am the PMRF Fellow that is hosting these sessions on Tuesday. As you would have already checked it out, but here we have the YouTube playlist on which all the live recordings of all of these sessions are being uploaded, so you can watch it at your leisure at a later point of time to clear any doubts you have, or if you have not been able to attend the live session, then you can just go through this here. So firstly, I would like to take any questions regarding the lectures of this week, if you have any that you are confused about, or you want more clarification on, then we can move on to the assignment questions from this week. So does anyone has any questions regarding this week's video lectures on NPTEL that were uploaded? Okay, so I guess not. So we will be diving directly into the assignment questions, in which we will be dealing with the questions first, then trying to answer them in the chat, and then discussing about them. So the first question of today is, the energy of UV light is blank. The options are lower than visible light, higher than visible light, equal to visible light, and none of the above. You can put whatever you think is the right answer in the chat, and go via ABCD, whatever you think is correct. So Vishwajit and Trupti both say B, higher than visible light. Aruna says A, lower than visible light. Jagruti, Shanti Priya, both of them saying B, higher than visible light. Lisa, Priyanka, and Mona Lisa, all three of them say higher than visible light. Kamlesh and Lakshmi Priya also say it is B, higher than visible light. Bigyan also says it is B. Anyone else wants to answer? Namrata also says it is B, higher than visible light. Okay, so we have a lot of answers in the chat. Let us quickly go through it. So I think this is you probably have come across this particular equations before. This particular equation relates the energy of a photon packet. So photons, as we know, is the minimum, the smallest unit of energy that is present in light, which is actually an electromagnetic wave, and it is related to, directly proportional to the frequency of the electromagnetic wave. And if you want to make, and the proportionality constant here is Planck's constant. So E equal to h nu, where this gives us that energy of a packet of electromagnetic radiation is directly proportional to the frequency of the radiation. Next, how is frequency and wavelength of electromagnetic radiation related? The equation that it follows is c, which is the speed of light, equals to lambda time nu. So lambda is the wavelength and nu is the frequency. So as wavelength increases, frequency decreases. They are inversely proportional. This follows that energy and wavelength are also inversely proportional in nature. So energy and frequency directly proportional, energy and wavelength inversely proportional, and wavelength and frequency are also inversely proportional. So now, what is the wavelength of the frequency of normal visible light as opposed to UV light? So here we have the entire electromagnetic spectrum, starting all the way from gamma rays, followed by X-ray, UV rays, a very small amount of the visible spectra, followed by infrared, microwaves, radio waves, and long radio waves. As you see right here, from left to right, the wavelength of the radiation increases. The wavelength of the light rays increases and the frequency increases from right to left. This follows that UV light will obviously have a higher frequency and a lower wavelength compared to visible light, as you can see right here. Visible light has a lower frequency and a higher wavelength, which tells us that according to the previous equations, that visible light should have a lower energy than UV light because its frequency is lower. Here we have examples of different types of uh, different types of radiation when they are produced. So gamma rays are majorly produced during nuclear transitions. X-rays are produced when core electrons transition amongst themselves. UV is produced when valence electrons transition amongst themselves. Visible also is when valence electron transition upon themselves. IR is when molecular vibrations are present. Microwaves are when molecular rotations are taking place, and radio waves are when nuclear spin is taking place. So here I would like to impart you with the knowledge that this in between UV, visible and infrared are directly related to the valence electrons and molecular vibrations, and thus we have actually developed multiple techniques that depend on the radiation that comes from the molecules during valency transition or molecular vibrations. Can anyone give me an example of such a technique that we have utilized to actually figure out the various transitions between electrons, or a technique that we use to actually figure out what are the bonds between different molecules? Does anyone have any idea? Okay, so there is something known as UV visible spectroscopy that is used to actually figure out the concentration of different solutes, which actually follows the same idea here, that you throw UV ray, you throw visible light into a particular solution, and depending on the absorbance of the sample, we can figure out what the concentration or what the amount of solute is present in a solution. There is another technique known as IR spectroscopy, in which we figure out the bonds between various molecules. So, say OH bond and a carbon bond will give a different IR spectroscopy data as compared to a double bond between two carbon atoms. So we have essentially looked into all of these transitions and how exactly these rays are formed and based various interesting and important techniques on them. Okay, so if that is clear, then if we go back to our question, the energy of UV light is higher than visible light. Is that clear with everyone, or does anyone have any questions from this? Okay, I hope that is fine. Moving on to the next question, which is also similar. The wavelength of visible light is blank and the frequency is blank than UV light. So as compared to visible light, the wavelength and frequency of UV light is, options are lower and lower, lower and higher, higher and lower, and higher and higher. So you have four options, A, B, C, D. Whichever you think is the correct one, you can put into the chat. Trupti says it is C. Wavelength of visible light is higher and frequency is lower than UV light. Kamlesh also says it is C. Lisa also says it is C, higher and lower. Shantipriya Jagruti also say it is C. Mona Lisa and Namrata also say it is C. Anyone else? Vishwajit also says it is C. Okay, so we have a lot of C's, all of us saying that wavelength of visible light is higher than UV light and the frequency is lower than UV light. Okay, so again looking at the electromagnetic spectra, here we have the frequency rising from left to right, so essentially the image is inverted as opposed to the previous one, and the wavelength decreasing from left to right. As you can see right here, visible the frequency of visible light is around 10 to the power 15 hertz, whereas that of ultraviolet light is around 10 to the power 16 hertz. Similarly, the wavelength is 10 to the power minus 6, two orders higher than the ultraviolet. Thus, as we have seen in the previous discussion as well, ultraviolet has a higher frequency and higher energy as compared to visible light. Similarly, our visible light has a higher wavelength as compared to ultraviolet light. Thus, the wavelength of visible light is higher. Okay, there is some, so the answer is marked wrong. It should be lower. The wavelength of visible light is higher and the frequency is lower. Okay, huh. So you guys are correct. I was getting confused for a bit. Yes. So the wavelength of visible light is higher and the frequency is lower as compared to UV light, because obviously the energy content of visible light is lower than that of UV. That is why the frequency is also lower and the wavelength is higher. Is that okay with everyone? You can put a thumbs up in the chat if that's fine. Okay, Trupti says it's fine. Kamlesh also says it is fine. So next we have a different type of a question. Which of the following which of the following is not a polymer? The options are DNA, dextran, RNA, and sodium. So we have flurry of answers. Trupti, Lakshmipriya, Jagruti also it is D, sodium. Mona Lisa, Shantipriya, Kamlesh also say D. Sunita, Shivani, Namrata, Shivani again, and Aruna also says it is D, sodium, and Shraddhanjali also says it is D, sodium. Okay, so quickly going through this. So this, can anyone tell me what this polymer is in the chat? We have gone through this earlier in one of our sessions. Does anyone remember what this exactly is? Just to describe it a bit more, this has a phosphate backbone, has a pentose sugar in it, and a lot of nitrogenous bases. This would then be which polymer? Any guesses? Even like you can even guess in the chat box whatever you think. Screen is not visible. Okay. Is the screen visible now? The screen is visible, right? Okay. So any guesses on what this polymer is? A long chain of, think of the molecule DNA. Excellent. Aruna and Monalisa both said DNA. Yes, you are correct. This is a DNA polymer with a phosphate backbone and a ribose sugar as well as four different nitrogenous bases. So these bases, depending on the structure, there are four bases present in, I will remove this, present in DNA and four bases present in RNA. The four bases present in DNA are adenine, guanine, cytosine, and thymine, whereas the four bases present in RNA are adenine, guanine, thymine, and uracil. So, in case of cytosine and uracil, in case of DNA, we have thymine present, no uracil. In case of RNA, we have uracil present, no thymine, and they essentially go in this location, shown right here on top of the ribose sugar. And does anyone know what is the difference between a DNA polymer and a RNA polymer is? What exactly would be the difference between if we had a similar RNA polymer right here with the same nitrogenous bases? Other than the bases, what is the difference between RNA and DNA? Yes, Aruna, you are exactly correct. So, as the name follows, the RNA is ribonucleic acid and DNA is deoxyribonucleic acid. So, as the name follows, there is a removal of an oxygen atom in DNA. So, in case of RNA, there would be a OH present here, right here, in the second position of the ribose sugar. RNA, there will be a OH present here. As Aruna has said, Shraddhanjali also says the same. You are absolutely correct. And this removal of a OH actually makes the DNA much more stable as compared to its RNA counterparts. The reason is because RNA degrading enzymes can target this particular OH, as well as RNA by itself also undergoes self degradation by hydrolysis using this particular OH, which is absent in case of DNA, and thus we see that DNA is much more stable than RNA. Okay, so this was about DNA and RNA. Moving on, we have another polymer here. This is dextran. Dextran is a polymer of a carbohydrate which forms a long chain. So as opposed to DNA or RNA, which has a particularly complicated monomers which forms long chains, dextran is made out of the same thing. This six-sided ring, as you can see, it repeats, and this can bond either via this oxygen or via this oxygen. So if it bonds in the oxygen which is at one end, which is known as a a 1 to 6 bond, then it forms this long chain. If it forms a 1 to 4 bond, then it forms a branching in the chain. So you can see that this is a branch that is shown right here, and this is the main long chain. So dextran is a polymer of a very simple monomeric carbohydrate. Does anyone remember what the monomer of dextran is? You can see it right here, but what is the name of this structure? The hint is that it is the most common carbohydrate ever. A six-sided carbohydrate, which is a monomer and the most common carbohydrate or sugar present in the world. Another hint would be that this particular molecule is made in photosynthesis. Yes, Kamlesh, Surajit and Namrata, you are all correct. This dextran is a polymer of glucose, and glucose in its cyclic state looks like this. This forms a six-membered ring with an oxygen in middle of it. This is how glucose is formed, and we know that glucose is formed during photosynthesis as well, and causing it to be the most abundant carbohydrate present in the world. So dextran is a polymer of glucose. Okay, so going back to our question, which is now after all of the discussion should be very simple. Which of the following is not a polymer? DNA, dextran, RNA or sodium? The answer is sodium. We have already seen DNA and RNA are polymers of ribonucleotides and deoxyribonucleotides, and dextran is a polymer of glucose. Okay, another question following the DNA. If you have been paying attention, so you should be able to answer this as well. Which of the following are found in DNA? Adenine guanine thymine cytosine, adenine guanidine thymine cytosine, adenine guanine uracil cytosine, adenine guanidine uracil and cytosine. We have a lot of answers again. Jagruti, Lakshmi Priya, Aruna say A. Trupti, Mona Lisa, Shanti Priya also say A. Priyanka, Shraddhanjali, Namrata also say A. Sunita, Bigyan, Kamlesh, Shibani, and the final person is again Shibani. All of them say A. So everyone seems to have been paying attention with hawk's eyes. So you all say that it is adenine guanine, thymine and cytosine. So we have just gone through it. You are correct that in case of DNA, the four bases are adenine, guanine, thymine and cytosine. In case of RNA, thymine gets replaced by uracil. So you are correct. The correct option is A. Just for your information, guanidine and thymine. So guanidine is the nucleoside of guanine. So the base, just the base, is known as guanine. If it is present along with a ribose sugar, it is known as guanidine. Similarly, thymine is the base, thymidine is when it is associated with the ribose sugar. Okay, moving on. In the next question, in a cell, concentration of blank is higher outside the cell and concentration of blank is higher inside of the cell. The option are Na minus, K minus, Na plus, K plus, K plus Na plus and K minus Na minus. Okay, so Aruna, Shraddhanjali, Mona Lisa all of say it is B. Trupti, Jagruti, Priyanka, Lakshmi Priya also say it is B. Shanti Priya, Shibani, Sunita, Vishwajit, Surajit, Kamlesh and Namrata, all of us say it is B, that the concentration of sodium is higher outside the cell and the concentration of potassium is higher inside the cell. So you are absolutely correct. We'll just quickly go through the discussion. So if we look at the concentration differences of various ions extracellularly and intracellularly, we will find something interesting. We'll find that depending if we are looking at the extracellular side or the intracellular side, there is a difference in concentration of these ions. There is a huge quantity of potassium present inside the cell as opposed to the outside, and a huge quantity of sodium and chloride present in outside of the cell as compared to inside. So this is something that the cell has to, we observe this, but obviously we know according to the second law of thermodynamics that such disparity cannot exist naturally in the environment. You have to physically spend energy in order to maintain such a disparity, and this is done via something known as the sodium potassium pumps which are present in the cell membranes. These sodium potassium pumps are specific in direction as well as substrates. They allow only potassium ions to go into the cell and push sodium ions outside of the cell in the by using ATP, the energy currency of the cell. In this way, the balance of sodium and potassium, this irregularity, is maintained across the cell membrane. So there are two questions that can be posed here. First, why do you think that there cannot be, like you cannot maintain such a disparity in nature? Like, why do you have to spend energy to maintain this disparity? Does anyone have any idea about this? Like, say, why do we say that this kind of a situation cannot exist? Why do you have to spend energy to maintain it? Anyone have any guesses about this? Yes, this is against the concentration gradient. And so Arun, another question is that there is a lot of potassium present inside the cell and very little outside the cell, and the vice versa in case of sodium and chloride ions. But why is this something that needs energy to be maintained? So Surajit and Trupti has said that this is because there is a concentration gradient that is formed. So there is a huge amount of potassium inside the cell as compared to the outside of the cell. So all the potassium wants to go outside. So it is against the concentration gradient, and always molecules move from a higher concentration to a lower concentration. And that is why when you want to maintain it in the opposite direction as the cell wants to, we have a lot of energy expenditure in order to do it. But there is also an interesting notion here that why does everything wants to move from a higher concentration to a lower concentration? What force actually causes them to move from higher concentrations to lower concentrations? Can anyone answer this? This is a bit more complicated, I would say. and needs a bit more understanding on your part. But I think you can answer this if you have an idea. Why exactly do concentration gradients not exist naturally in the environment? Always something wants to move from a higher concentration to a lower concentration. Never will you see, without the expenditure of any energy, that there is a unnatural concentration gradient being maintained. Why is that? Electrostatic force. Not in this case, not really, because both the ions are similarly charged. So there is not much question of electrostatic force coming into play. So I will give you the answer. I will briefly give you the answer. You can actually think about it and come back next day and try and discuss this. So in thermodynamics, there is a concept known as entropy. Entropy essentially is the measure of how much randomness there is present in a particular sample. So, and entropy is obviously directly related to the chaos in a sample. So, how many forms or how much randomness there is present in something. So, see, in the situation where you have a warm tea, the molecules of the tea are moving around everywhere at random because they have a lot of kinetic energy. So that is a high high entropy system, versus cold water where the molecules are lethargic, do not move around a lot because they do not have a lot of kinetic energy. That is a low entropy system. The second law of thermodynamics states that the entropy of the entire universe is always rising. Everything wants to go from a high entropy from a low entropy state to a high entropy state. So, from a high entropy state to a low entropy state. So if you actually go through these concepts a bit, you will realize that this particular situation is a highly entropic situation because you are maintaining a disparity. So this is not random, this is actually forcibly being maintained here. This in turn actually causes electropotential gradient, as Shraddhanjali has said, which in turn causes the solutions to mix. So always our every kind of solution or every kind of substance in the entire universe always wants to go to a lower entropy state, and that is why all of this happens. So that is a lot of thermodynamics. Essentially, uh, cutting it short, when there is a concentration gradient, always everything wants to move from a higher concentration to a lower concentration because of this, and that in turn is caused by electrostatic forces or electropotential gradients or concentration gradients of the chemical nature. So a lot of talk, but after this we go back to a question. In a cell, dash concentration of dash is higher outside the cell and concentration of dash is higher inside of the cell. So the answer is sodium and potassium. Is that fine with everyone? Okay. Yes, we go back to our sodium potassium pumps. Uh, the sodium potassium pump exchanges blank amount of potassium and blank amount of sodium. So Aruna says it is C, 3 and 2. Jagruti says it is D, 2 and 3. Mona Lisa says it is C, 3 and 2. Trupti, Shanti Priya, Shraddhanjali say it is D. Surajit, Lakshmi Priya, Kamlesh say it is D. Priyanka, Mona Lisa and Sunita also say it is D. Namrata says also that it is D. That it is 2 potassium in exchange for 3 sodium. Okay. We'll be looking at the sodium potassium pump because this will be coming up in our discussion regarding membrane potential. So we'll quickly go through this. So the sodium potassium pump essentially exchanges 3 sodiums in exchange for 2 potassium ions, and that is powered by ATP. So this is the entire phenomenon that occurs. First, in the sodium potassium pump is facing towards the intracellular side. So it is facing towards the cytoplasm. Two events occur. First, ATP binds to the pump and it catches hold of 3 sodium ions. Once it catches hold of 3 sodium ions, the entire ATPase pump utilizes the ATP and gets phosphorylated. So it now has a phosphate attached to it. Once the phosphate is attached, the entire pump changes its structure and releases the sodium ions to the outside of the cell. Now this pump is open towards the extracellular side and closed towards the intracellular side. In this situation, 2 potassium ions bind to the pump. Once 2 of them are bound, the phosphate that was added by ATP is released and the pump goes back to its original intracellular facing side. So it now faces intracellularly. Once the phosphate is released, this potassium are also released into the cytoplasm. So we have exchanged 3 sodium, kicking them out and taking in 3 potassiums. And now the entire cycle begins again. ATP binds to the pump and 3 sodium are attached to it. So again the same cycle will repeat. Sodium going out 3 molecules and potassium coming in 2 molecules. Is that fine? Any questions from this? Okay. So moving on, the sodium potassium pump exchanges 2 potassium in replacement for 3 sodium. The answer is D. Okay. Next, the axon hillock is an blank structure and the synapse is a blank structure. The options are interneuronal, 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 intraneuronal, intraneuronal, interneuronal, and intraneuronal. So intra meaning in the same neuron, inter meaning between two neurons. Okay, so with the answers coming in, Trupti, Mona Lisa, and Shantipriya say it is C. Lakshmi Priya, Shraddhanjali, and Kamlesh say it is C again. Mona Lisa, Banita, and Vishwajit also say it is C, which is axon hillock is intraneuronal and synapse is interneuronal. And Vishwajit, Surajit, Jagruti, and Shibani also say it is C. Axon hillock is intraneuronal and synapse is interneuronal. Okay, so we'll quickly go through the structure of neuron. So this is how the exact neuron looks like. It is what we know as a polarized cell. A polarized cell has two ends which look very different from each other, and this is very evident in case of neurons. Aruna says it is not. Is What do you mean, Aruna? Is my screen not visible, or is there any question regarding this? Okay, okay. So coming back to this, neuron is a polarized cell with two different, very different looking two arms or two different looking parts. One on one end, the neuron has its nucleus and the entire cell body, which is the left part. So you can see the nucleus, and from the cell body there are many processes which are extensions of the entire cytoplasm which branch out like this. These smaller processes are known as dendrites, and this entire formation is known as the dendritic tree of the neuron. But on the other end, the neuron has one process which is especially long. This process is known as the axon, and the axon on its end also has various connections like this. So the neuron, for your knowledge, it acts as a unidirectional messenger. It receives messages on this end, on the dendritic end, and sends its messages via this axonal end. So here there are various parts to the entire neuron. If we look closer, this neck-like region where the axon is connected to the nucleus or the soma is known as the axon hillock. This particular part is full of but important kind of ion channel. A ion channel is present on the membrane and allows for either coming in or going out of various ions. The ion channel present here in abundance at the axon hillock is the voltage gated sodium channel. So ion channels can be of three types. This is a lot of information. Please stop me if you cannot understand something. So ion channels are ones that allow the transfer of ions through the cell membrane, because ions cannot go through the lipid bilayer of the cell membrane and need a pathway which the ion channels provide. These ion channels can be of three types. First is a ligand gated ion channel. When a ligand, which could be a hormone, which could be a neurotransmitter, which could be a protein, which could be a lipid, bind to the external surface of the channel opens and allows transfer of ions. A second type of ion channel is a voltage gated ion channel. Depending on the charge present on the cell membrane, the voltage gated ion channel can either open or it can close. And the final is known as a mechanical ion channel, which opens or closes depending on the stress or pressure present on the cell surface. So the axon hillock has abundance of voltage gated sodium channels, which actually allows the signal transmission to go from a dendrite to axon direction, from a left to right direction. We'll see how that happens in the upcoming slides. So this is known as the axon hillock. Further, the entire neuron is covered, or the axon of the neuron is covered in another layer of cells. These cells are known as Schwann cells, and a lot of them come together to form what is known as the myelin sheath present on the neurons. These essentially act as insulators, causing which do not allow another neuron which is close by to actually transfer signals horizontally like this. It can not go like this in this fashion, but only can go from a left to right fashion. Only horizontal transmission, no vertical transmission via the axon. And finally, at the end of the axon, there will be another neuron present whose dendrites will be right here at the end of the axon. This structure looks something like this. So before we move on, is there any questions regarding the anatomy of the neuron? Because we will be moving forward with a lot of detail about this. And if anyone has any questions, you can just tell me. So giving you a minute here to go through this. If there is no problem, then we'll go to the next structure. So axon hillock, as you can see, is a intraneuronal structure. It is present inside the neuron. At the ends of the neuron, both here at this terminal as well as this terminal, the neurons are in connection with another neuron. But the axon is always facing towards another dendrite. The axon is someone that gives the signal, dendrite is someone which receives the signal. So, and they together form a structure known as the synapse. So here we have the axonal terminal in the top, and here we have the dendritic terminal at the bottom. So the synapse, there are two types of synapses. There is one known as a chemical synapse that is shown here, another known as an electrical synapse. We will be discussing the chemical synapse today. So how exactly is the signal transferred from one neuron to another? So there is a very interesting bit of biochemistry going on here. So there are, as you can see, the so this is the axonal synapse is known as presynaptic. The dendritic part of it is known as postsynaptic. Before the synapse, after the synapse. The presynaptic terminal contains many vesicles containing a substance known as neurotransmitters. So you can see these as the dots present inside the vesicles. Once the axon is ready to send a signal to the next dendrite, these vesicles fuse with the cell membrane of the axon and release these various neurotransmitters into the synaptic cleft, the very small region between the two neurons. These neurotransmitters then go ahead and bind to ion channels, which are ligand gated ion channels present on the postsynaptic dendrite. Once these ion channels are bound by the neurotransmitters, these open up and allow transfer of ions. And these transfer of ions cause the signal transduction to the next neuron. So once these channels are open and there is transfer of ions, that is the signal that causes the next neuron to fire or actually have an action potential. So this is how exactly synaptic transmission works. Any questions from this? Okay, so it says no. I hope this is clear with everyone. So moving forward, difference between synapse and synaptic cleft. So the entire structure formed by the presynaptic terminal and the postsynaptic terminal, including parts of the both cells and the ion channels and the neurotransmitters, everything that you can see in the picture, this entire thing is the synapse. The small cavity between the two neurons is known as the synaptic cleft. So it is like if my two fingers are here, the two fingers are the two neurons. So the part containing the two fingers, the end of the two fingers is the synapse, and the space in between the two fingers is the synaptic cleft. Is that fine, Aruna? Okay. Someone raised a hand. I think it was Vishwajit. Do you have any questions?
Mm,
Okay. I guess that was a no. Okay. So yeah. The axon hillock is an intraneuronal structure, and the synapse is an interneuronal structure. Does anyone else have any questions from this? Okay, moving forward. Now we have delved directly into how exactly neurons work, and we'll be going on to the first question about working of neurons. The question is, resting membrane potential of a neuron is blank, and the negative sign indicates there are more blank outside the cell than inside the cell. So the options are minus 70 positive ions, minus 70 negative ions, minus 10 positive ions, and minus 10 negative ions. So Jagruti, Trupti, and Lakshmi Priya, all of them say it is minus 70, the resting potential, and positive ions that are more outside of the cell. Mona Lisa, Shanti Priya, Mona Lisa, Banita, and Surajit also say it is A, the resting membrane potential of a neuron is minus 70, and the negative sign indicates that there are more positive ions outside of the cell than inside of the cell. Anyone else wants to answer? Okay, here we have a video that can explain everything to us easily, so we'll be moving to that and discuss again. So this should be the video. This screen is not visible. Yes, I have not shared it yet. Is this visible now? Just let me know. I will remove this here. Let me know if this is audible.
In this video, I want to talk about how the neuron resting potential is created.
This is audible, right?
And how it relates to concentration differences.
So we'll go through this video now.
In this video, I want to talk about how the neuron resting potential is created and how it relates to concentration differences in some of the important ions involved in neuron function, because understanding the concepts involved in the neuron resting potential will help us understand other neuron membrane potential changes like the graded potentials and the action potentials. So here I've drawn kind of a blown up neuron. So we'll have the soma in red, the axon in green, and one dendrite in blue, and I've blown up the dendrite and the axon really large just so I have some space to draw. It's useful to consider the formation of the neuron resting potential in imaginary steps, but in reality they all occur simultaneously. So first, let's consider a neuron with no resting potential, so it's not more positive outside or more negative inside the membrane, and we'll consider that all these key ions have the same concentration inside and outside the neuron, so that there are no concentration gradients for organic anions, potassium, sodium, chloride, or calcium. Now the neuron is going to be creating organic anions and adding them to the cytoplasm. And as organic anions are being created, most of which are proteins with a net negative charge, these extra negative charges that are being added to the inside of the membrane are going to create a small membrane potential, so that now it'll be more negative inside the membrane of the neuron because of these extra negative charges. But this will be something small, like maybe it'll be somewhere around -5 millivolts. I don't actually know what this would be, but it's going to be some small membrane potential that's not going to be enough for the neuron to function. But even with a small membrane potential, there will now be an electrical force acting on the organic anions, because they will be attracted to the more positively charged outside of the membrane, so that the electrical force will be trying to drive organic anions out of the neuron, but they won't be able to leave, because it turns out that the membrane is highly impermeable to the organic anions. And the same will be true for the diffusion force caused by the higher concentration of organic anions inside the neuron compared to outside the neuron. Even though these electrochemical driving forces are trying to make the organic anions leave, they can't get past the neuron membrane, so they're trapped inside the neuron. So no changes will happen to the concentrations of the organic anions, and no further changes will happen to the membrane potential at this imaginary step. The same is not going to be true for the other ions, because the neuron membrane has channels in its membrane called leak channel or leakage channels. And these channels allow these ions to pass across the membrane, although how easily these ions can cross the membrane through the leak channels is very different for the different ions. And these leak channels are open all the time. They're not gated, so they're not opening and closing in response to some kind of stimulus. For the next imaginary step, let's consider what happens when we add the sodium-potassium pump to the neuron membrane. So this is going to be our sodium-potassium pump. This will be an active ion transporter. It will use the energy of one adenosine triphosphate molecule, that I'll write as ATP for short, to actively transport 3 sodium ions outside the neuron, in exchange for 2 potassium ions being transported to the inside of the neuron. Now, by moving more positive charges outside the neuron than it's moving inside the neuron, that's going to make the membrane potential bigger. It's going to make it more negative inside the neuron membrane. I'm not sure exactly what size of change that would be, but it's going to be something small. Let's just say, for example, it might be around negative 10 millivolts. So now we have a bigger membrane potential, but it's still probably too small for the neuron to function. Far more importantly than this change to the membrane potential, however, is that we're going to have a big change to the concentrations of potassium and sodium inside the neuron. By pumping potassium inside the neuron, we will now get a much larger concentration of potassium on the inside of the neuron than we have on the outside of the neuron. And by pumping sodium outside of the neuron, we're now going to have a much smaller concentration of sodium on the inside of the neuron compared to the concentration of sodium on the outside of the neuron. Now, you may be wondering why the concentrations of potassium and sodium changed inside the neuron, but they didn't change on the outside of the neuron. And the reason for that is that the extracellular fluid, all of this fluid outside of the neuron, is huge, and the total number of ions outside the neuron in the extracellular fluid is very large compared to the volume of the cytoplasm inside the neuron and the total number of ions inside the neuron. So that any of these movements of ions across the membrane of a neuron is really going to only change the concentration of ions inside the neuron, and any change to the concentrations of ions in the extracellular fluid is going to be negligible. It's going to be so small it's not even worth considering. There are now opposing forces on the potassium ions. The electrical force will be trying to drive these cations in to the more negative inside of the cell, but the diffusion force will be trying to drive potassium out of the cell from a higher concentration to a lower concentration. At typical neuron ion concentrations at this imaginary step, the diffusion force would actually be much larger than the electrical force. So while there are forces pointed in opposite directions acting on potassium, the net electrochemical driving force, that is the larger diffusion force minus the smaller electrical force, will cause a net movement of potassium ions out of the neuron through the leak channels. As potassium ions are leaving the neuron and carrying their positive charges out of the neuron, that is going to make the inside of the neuron membrane more negative. The membrane potential is going to get bigger and bigger with each potassium ion that leaves. And it's going to continue to do so until the membrane potential is big enough that the electrical force trying to drive potassium ions into the neuron is the same size but in the opposite direction as the diffusion force trying to drive potassium ions out of the neuron. When these forces are the same size and pointing in opposite directions, there is no net movement of potassium ions into or out of the neuron. The occasional potassium ion will wander out or wander into the neuron, but in basically even amounts. For potassium, at typical neuron ion concentrations, this might occur around -70 millivolts, which is more than enough of a membrane potential for the neuron to function. The membrane potential, where an ion has balanced electrical and diffusion forces so that there's no net movement of that ion across the membrane anymore, is called the equilibrium potential. It's also called the reversal potential. Now, it actually doesn't take that many potassium ions to exit the neuron to reach the equilibrium potential for potassium. It's something like less than 1% of all the potassium ions in the neuron have to leave for potassium to reach its equilibrium potential, so that the effect on the intracellular concentration of potassium is really negligible. This process does take a little time, though, because most of the membrane is impermeable to potassium, and it can only squeeze out through the leak channels. Now, for sodium ions, both the electrical force and the diffusion force are strongly trying to drive sodium into the neuron, because it's a cation, so it's attracted to the negative inside of the neuron membrane, and its concentration is much higher outside the neuron than inside the neuron. If we had a membrane that was only permeable to sodium, these cations would flow into the neuron until so many positive charges entered the neuron that it actually wouldn't be negative inside anymore. It would actually become positive, at which point the electrical force would switch, and it would have to get quite positive inside the neuron for the electrical force to get big enough to balance the diffusion force to reach the equilibrium potential of sodium, which at typical neuron ion concentrations may be around positive 50 millivolts. Without input, however, when the neuron membrane is at rest, the permeability of the membrane to sodium is actually quite a bit less than it is for potassium. And in fact, the permeability of the resting neuron membrane to sodium is something like 4% of that of potassium, so quite a bit less. But because some sodium is able to squeeze through these leak channels, bringing some positive charges into the neuron, that is going to affect the membrane potential a little bit, so that instead of being at the potassium equilibrium potential of around -70 millivolts, at this imaginary step it might settle in around -60 millivolts. And that's actually a pretty typical neuron resting potential. When the membrane is permeable to multiple ions that have electrochemical driving forces, the resulting membrane potential is a weighted average of the equilibrium potentials of those ions, weighted by their permeability. At rest, the neuron membrane is much more permeable to potassium ions than it is to sodium ions, so that the resting potential is much closer to the equilibrium potential of potassium than it is to that of sodium. Because the concentrations and permeabilities of ions are usually stable in most resting neurons, the resting potential is usually stable as well. Now, neither potassium nor sodium is at its equilibrium potential, however, so there will be a small amount of net movement of both ions across the membrane. A little bit of potassium will be dribbling out of the membrane, and a little bit of sodium will be dribbling into the membrane. This will be matched by ongoing activity of the sodium-potassium pump, which not only creates these concentration gradients in the first place, but also maintains them over time. The resting membrane usually has an intermediate permeability to chloride ions of around 45% of the permeability to potassium ions. In contrast to potassium and sodium, whose concentration gradients play a big role in creating the resting membrane potential, for chloride, the resting membrane potential plays a big role in determining its concentration gradient. The membrane potential drives chloride out of the neuron until its concentration gradient is big enough to balance it, so that normally there's a very small concentration of chloride ions inside the neuron compared to the outside. And because of that, the equilibrium potential for chloride is usually at or close to the typical resting potential for a neuron of around -60 millivolts. Most neurons have other active means of decreasing the intracellular concentration of chloride, though, the main one being the chloride-potassium symporter. And this drives chloride out of the neuron by harnessing the diffusion force acting on potassium ions, So it lets potassium move out of the neuron like it wants to, and it harnesses that energy to also push more chloride ions out of the neuron, further decreasing the intracellular concentration of chloride. Because of this, the equilibrium potential of chloride for most neurons is usually not at the resting potential of around negative 60 millivolts, but instead it's around negative 70 millivolts, which will cause some inward chloride flow, leading to a minor change in the resting potential to a more negative value, although usually the change is negligible. Most neurons also have active means to decrease the intracellular concentration of calcium, so that there is a small concentration of calcium ions inside the neuron compared to the outside of the neuron. The main one of these being the sodium-calcium exchanger, which I'll show right here, that drives calcium out of the neuron by harnessing the energy of the electrical and diffusion forces acting on sodium, so that the calcium-sodium exchanger allows sodium to enter the neuron in exchange for pumping calcium out of the neuron. And this creates strong electrical and diffusion forces trying to drive calcium into the neuron. And these forces are so big that the equilibrium potential of calcium wouldn't be reached until the inside of the membrane was around positive 120 millivolts to flip the electrical force to balance the diffusion force trying to drive calcium into the neuron. But it turns out that the resting membrane permeability to calcium is actually quite small, so that it usually has little effect on the resting membrane potential. So while the concentration gradients of chloride and calcium ions tend to have little effect on the resting membrane potential of most neurons, they do play major roles in other aspects of neuron functions, which we'll talk about in later videos.
Okay, that was a very interesting video, I hope. Does anyone have any questions from the video? I think we have gotten a lot of information from the video itself about how exactly the resting membrane potential of a particular neuron is maintained. Any questions from this? Okay. Okay. In case of different ionic species such as sodium, potassium, calcium or chloride, we have seen that the permeability of all of them as well as the concentration of them depends on the resting membrane potential. So there were three scientists, Goldman, Hodgkin and Katz, who figured out a way to actually or gave a way to calculate the mathematical formula for membrane potential. So generally how membrane potential is recorded is that they have a voltmeter kind of apparatus which they insert inside the neuron, and another end rests outside the neuron in order to measure the membrane potential, which comes around to, as you saw, minus 70 or minus 80. But what about the mathematics behind it? This was given by the Goldman-Hodgkin-Katz equation, in which this is what the formula looks like. Em, or the membrane potential, equals to RT by F log of the permeability of a particular ion multiplied by the concentration of the ion outside of the cell and divided by the permeability of an ion divided by the concentration inside of the cell. And this will give you a similar value to what you will measure on your voltmeter. Okay, coming back all the way back to our question, which is resting membrane potential of a neuron is blank. The negative sign indicates there are more blank outside the cell than those inside the cell. The correct option was resting membrane potential is minus 70 and there are more negative ions outside the cell. No, this The answer is here is a bit wrong. It will be there are more positive ions outside the cell than inside the cell. You all are correct. I have marked this mistakenly wrong. Okay. Any questions from either the video or the question? Anyone? Anyone wants to ask anything? Okay. Moving on. Redistribution of electric charges of a cell occurs. It leads to less negative charge inside the cell than outside. This will be called as hyperpolarization, polar peak, depolarization, or sinoatrial faulting. So 4 options A, B, C, D, whichever you think is the correct one you can put into the chat. Aruna says it is C, depolarization. Inside becoming less negative is depolarization. Surajit also says it is C. Lakshmi Priya says it is B, polar peaking. Anyone else? Lakshmi Priya changes her answer to C, or depolarization. Kamlesh also says it is C, depolarization. Shraddhanjali also says it is C. Mona Lisa also is saying this is C. And Shanti Priya also says that is C, depolarization. Okay, let's quickly go through this. So we here have another discussion on action potential mechanism of neurons. So we'll go through that. Yeah, now the video for you. Let me know if this is audible.
In this video, I want to talk about how action potentials are generated at the triggers.
This is audible, right?
Zone and how they're conducted down the
audible, right?
axon. So I've drawn a
Everyone can hear this? Hello? Okay.
a soma here in red and one axon in green, and I've blown up the axon to a very large size just so I have some room to draw. Here's our graph of the membrane potential on the y-axis and time on the x-axis. And now I've put a couple of different kinds of ion channels in the membrane of the axon. The first, in this lighter gray, are the leak channels that we talked about when we talked about the neuron resting potential. These channels are open all the time. They're not gated. And I have not drawn any ligand-gated ion channels like the neurotransmitter receptors that occur on the soma and the dendrites. But to talk about the action potential, I need to introduce an entirely new type of channel that I've drawn in dark gray with this little V, and these are voltage-gated ion channels. The membrane of an axon has many voltage-gated ion channels, most of which open when the membrane potential crosses a threshold value. So we've talked about the threshold potential before, and all of these numbers may vary between different types of neurons, but these would be fairly common values. So many neurons would have a resting membrane potential of around -60 millivolts, and a threshold potential of around -50 millivolts or so that I've drawn with a dashed line. And the importance of this threshold potential is that it determines if these voltage-gated ion channels will open. So when there is enough temporal and spatial summation of excitatory graded potentials to get us toward the threshold, here at the trigger zone, at the initial segment of the axon, so let me just draw that we have temporal and spatial summation of excitatory potentials spreading across the membrane of the soma into the initial segment of the axon, the trigger zone. This voltage-gated ion channel has a mechanism to sense this voltage change And when the threshold potential is crossed, it's going to open, and these are going to be sodium channels. Recall that the electrical and diffusion forces acting on sodium ions are strongly trying to drive them into the neuron. So when this voltage-gated sodium channel opens, sodium is going to flow into the neuron through the open channel, causing that part of the membrane to depolarize from all these positive charges now on the inside. This is going to cause an explosive chain reaction by triggering the voltage-gated sodium channels in the next piece of the membrane, so that more sodium is going to flow in, further depolarizing the membrane and opening the next voltage-gated sodium channel. These voltage-gated sodium channels open very quickly, triggering each other in a wave that rapidly spreads down the axon. The trigger zone has the greatest density of these voltage-gated sodium channels, which is why action potentials usually start at the trigger zone. So many of these voltage-gated sodium channels will open that the membrane permeability to sodium is dramatically increased. This is going to cause the membrane potential, which has already gone from the resting potential to the threshold potential from the graded potentials. But now that all this sodium is flowing in through these open channels, the membrane potential is going to dramatically rise, trying to head toward the equilibrium potential of sodium, which is usually somewhere around positive 50 millivolts. This rapid increase in the membrane potential values is due to these voltage-gated sodium channels. And this is called the rising phase of the action potential. And in fact, it becomes more positive inside the neuron membrane during this period. That it's the reverse of the resting potential, because normally it's more negative inside than outside the neuron membrane. But now so much sodium has entered that it's more positive inside the membrane than outside. The action potential usually peaks, though, somewhere around positive 40 millivolts. So it doesn't make it up to the sodium equilibrium potential that's often around positive 50 millivolts. And the reason for that is that these voltage-gated sodium channels automatically start to close at the higher potential values, so that sodium stops flowing into the neuron. And after they close, they're in a special state called the inactivated state, and they're unable to open at any membrane potential for a brief time. The next thing we see happen to the action potential, basically just as fast as the membrane potential went from the resting potential to the peak of the action potential, it then rapidly descends back toward the resting potential, and then actually goes farther. It goes more negative than the resting potential, and then it levels off. The reason for this part of the action potential, which is called the falling phase, is because potassium starts to exit the neuron. And it does so through a couple of types of channels. The first are the leak channels that we talked about when we talked about the resting membrane potential. Now, a little potassium is exiting through the leak channels at the resting potential, but even more potassium than normal starts to exit, because during these parts of the action potential, the membrane potential is positive, so that during this part of the action potential, both the diffusion force and the electrical force are strongly trying to drive potassium out of the neuron, so that more leaves through the leak channels than normally does during the resting potential. The second type of channel that allows potassium to exit are voltage-gated potassium channels. These also open when the membrane potential crosses the threshold, but they're a little slower to open than the voltage-gated sodium channels. So that first, all the voltage-gated sodium channels snap open, allowing sodium to rush in, causing the rising phase of the action potential, and then a little slower, the voltage-gated potassium channels open, allowing potassium to flow out of the neuron, contributing to the falling phase of the action potential. And then the action potential stops falling because now it's more negative inside the neuron again, so there's less driving force pushing potassium out through the leak channels, and also the voltage-gated potassium channels automatically close at the lower potential values, just like the voltage-gated sodium channels automatically closed. But just like the voltage-gated potassium channels were a little slower to open than the voltage-gated sodium channels, the voltage-gated potassium channels are also a little slower to close, so that it takes a little longer for this exit of potassium to stop. And that's why there's this little bit of a longer period at the end of the action potential till we kind of slowly settle back into the resting membrane potential. Because as these voltage-gated potassium channels are slowly closing, the membrane permeability to potassium is returning to the normal amount you get during the resting potential through the leak channels. And as that permeability to potassium returns back to the normal resting potential level, the membrane potential returns to the resting potential. This movement of sodium ions and potassium ions across the membrane, causing the waveform of the action potential, starts here at the trigger zone at the axon initial segment, but then rapidly spreads in waves down the axon. First, there's the wave of depolarization from opening of the voltage-gated sodium channels. So a wave of depolarization rapidly spreads down the axon. But following right behind it, right on its heels, is this wave of hyperpolarization caused by potassium exiting through the voltage-gated potassium channels and the leak channels. So we have the rising phase of the action potential, the peak of the action potential, the falling phase of the action potential, and then this period of hyperpolarization at the end of the action potential has a couple of names. It can be called the after hyperpolarization because it's the hyperpolarization that happens after this part of the action potential. But it's also called the refractory period. Let me just write that down. Refractory period. Refractory period. Right there. And it's called the refractory period because during this time it's difficult or impossible to trigger another action potential in that part of the membrane. The refractory period is divided into two parts. The first part is called the absolute refractory period, and it's absolute because the voltage-gated sodium channels, when they first close, they're in a special state called the inactivated state, and they are unable to open at any membrane potential for a brief time, so that no matter how much excitatory input comes into the neuron, you can't trigger another action potential during the absolute refractory period. The second part is called the relative refractory period, and during this time, the voltage-gated sodium channels have become functional again. They can respond to depolarization. However, the membrane potential is hyperpolarized. It's not yet back to the resting potential. Therefore, it would take more excitatory input than normal to trigger an action potential during the relative refractory period. One important effect of the refractory period is that action potentials travel from the trigger zone to the axon terminals, and they don't turn around and head right back the other direction because the membrane right behind the action potential is refractory. It can't be triggered by itself to send the action potential back the other way.
Okay, coming back, I hope that video was clear. Does anyone have any questions about action potentials? You can just ask me if you have any questions regarding understanding the last video. That was essentially our recap on how exactly action potentials work. We'll be discussing a bit on them. Okay, we'll quickly recap what they said in the video, so we will have a idea about it again. Is the screen visible? The screen is visible, right? Hello. The screen is visible, correct? Okay. So this, okay, this is the membrane potential by versus time graph that occurs during the action potential of a neuron. So we'll be understanding this via three different channels that are present on our neuronal membrane. The channels are a potassium channel, a sodium potassium pump, and a sodium channel. Here you have to understand that the sodium and the potassium channels are voltage gated ion channels. They allow the transfer of sodium and potassium only under when there is a change in the membrane potential, whereas the pump always works irrespective of the membrane potential via using ATP. So this is the resting situation where we have an initial stimulus, which generally is the transmission of neurotransmitters via the synapse, as we saw in our previous question, in which the neurotransmitter bind to the ligand gated sodium channels and open them up. This allows for a transient small increase or depolarization of the membrane to about -60 or -55 millivolts. Once this region is reached, which is known as the threshold membrane potential, the action potential now starts to occur. What happens is that once this threshold is crossed, the membrane potential has changed enough that the voltage gated sodium channels start opening one by one. Once the voltage gated sodium channels open, and we know there is a lot of sodium in the extracellular space as compared to inside the cell, there are a lot of sodium ions that start flowing from outside of the cell into the cell. This increases the positive charge inside the cell, contributing to further depolarization. So you see a huge amount, the peak continues to rise, and this continues to happen for a long time, or rather this continued to happen for a very short time, as you can see, between 1 to 2 milliseconds. So the sodium ion influx shoots up this entire voltage. So this is known as the depolarization phase. Next, what happens is that the the voltage gated sodium channels are very fast. So they open very fast and they close very fast as well. So once this peak is reached, the sodium channels have started closing. But there is a second channel that we had discussed, the potassium channel. The potassium channel is a slow channel. So due to the same stimulus of the membrane potential crossing the threshold potential, the sodium channels opened up. This same stimulus actually opens the potassium channels, but the potassium channels are very slow, so they will only open up around the time when the sodium channels are already starting to close, so around the peak. Once these open up, as we know that there is a lot of potassium inside the cell as compared to outside, the potassium starts to flow out in a rush. Thus there is a decrease of positive ions inside the entire cell, which causes the entirety of the inside of the cell to start getting repolarized. So positive going outside of the cell decreases the positive ion charge inside the cell. So we have negative coming back again. This negative actually is indicated by this decrease in the slope back to its original position. So this potassium channels uh, allow the potassium positive charge to keep on escaping till we have the entire slope going down a bit away from the initial value. So it goes below -70 to around -90. This is because the potassium channels are slow to close as well. So they close only after the negative charge has so hyperpolarization or increase in the negative charge as compared to the normal baseline has occurred. So it dips down below, and then again the ATP, the sodium potassium pump, starts working overtime to maintain and bring back the original situation or the original resting potential back again. So this is how the entire process occurs. Any questions from this? Okay. Since there are no questions, I assume everyone has understood. So the answer to this question: a redistribution in electric charges of a cell occurs it leads to less negative charge inside the cell than outside, it is called depolarization. When it is negative inside, it is called hyperpolarization, and if it is becoming positive, it is known as depolarization. Okay, so then this is the last question for today. For action potential to occur, the potential needs to shoot below minus 100 millivolts, shoot above minus 30 millivolts, remain between 60 and 70 millivolts, or remain at resting membrane potential. So the four options, you can put in whatever you think is the correct in the chat. Truptish says this is B, shoot above minus 30 millivolts. Surajit also says it is B, shoot above minus 30 millivolts. Anyone else? Bigyan also says it is B, shoot above minus 30 millivolts. Okay, so this is the diagram that we just saw. So here we see that the initial stimulus increases the membrane potential from resting membrane potential to a bit higher. So here the threshold is shown as minus 55, but depending on the neuron it could be minus 40, it could be minus 30. But once it crosses the resting membrane potential, then we have a huge depolarization, which is the action potential. So for this particular question, the only option that serves any purpose is shoot above minus 30 millivolt, which will be the initial threshold breaking stimulus, which will then cause the entire action potential to occur. Any questions from this? Okay. So that was all I had for you today. If anyone has any questions from today's lecture, then you can please let me know, then we can discuss it. Okay. So with that, thank you everyone for joining today's session. I will be seeing you next week with next week's session. Thank you.